we are starting today with another important topic, linkage and recombination. We know that there are many genes which actually decide the trait of an organism. For example, in human beings, approximately 30,000 genes are there, and the number of chromosomes is only 23. That means 46 chromosome, 23 pair of chromosomes are available. So all these genes are to be accommodated in a chromosome. The chromosomes therefore form the linkage groups. That is, linkage groups are all the genes which are present on one chromosome, and they together will form the linkage group. So suppose this is a chromosome, and the genes will be present on this particular chromosome at different loci, which we call it as gene loci. Now the genes are present on this, and this forms the linkage group. Now when we talk about the linkage, it is a phenomena in which two or more genes which are present on a chromosome are inherited together, and the frequency of their recombination is less than 50%. Let us understand this. Now when we are saying that the genes are present on the chromosomes, the genes are present at different locations. So certain genes which are placed close to each other. Now here what is actually happening is, suppose these are the genes. Suppose this is the gene and this gene. During the process of crossing over, let's say this is the I gene over here. Now, if we see this picture, we find that A and B are very close to each other, whereas A and I are very far from each other, although each of the gene is corresponding to a different trait. Now here, when the crossing over takes place between I and let's say O over here, so this two genes, since there is a lot of gap between the two genes, these can be separated during the crossing over, resulting into the recombination. So what is actually happening during the linkage, two or more genes which are present on a chromosome are inherited together. Here the possibility of A and I in being inherited together is very less, whereas the possibility of A and B over here, let me mark it with red also, A and B, since they are very close to each other, the distance between the two is very less as compared to distance between these two. As a result, the A and B gene can be inherited together, and these genes will be known as linked genes, and they will allow very less recombination, or the frequency of recombination will be very less. It will be less than 50%, whereas in this case the possibility of recombination, that is A and I, the possibility of recombination is very high. So linkage allows recombination depending upon the position of the genes. Now, depending upon whether it is allowing the recombination or not, the linkage types are of two types, complete and incomplete linkage. The complete linkage does not allow any crossing over. The crossing over is a process that happens between the non-sister chromatids of homologous chromosome, and it takes place during the prophase stage. So this crossing over which is happening over here, it is during the prophase 1 stage, during meiosis. that is the reductional division. So this crossing over allows recombination and also enables variation because it is during this period the genetic material is exchanged. Now, as a result, in the case of complete linkage, since it is not allowing any crossing over, that means over here, if we talk of gene A and B, it will not allow any crossing over and will always be inherited together. As a result, they will be showing the parental combination only. On the other hand, we have incomplete linkage in which it allows combination or crossing over and they are not necessarily inherited together always. The genes show variation. As a result, the offspring which will be formed due to incomplete linkage will show certain extent of variation. Now, all this depends upon the position of the genes. Here is another term which we have talked about much is recombination. Recombination is a process that allows exchange of genetic material during the crossing over. So this process which allows the exchange of genetic material over here is known as recombination and it produces the recombinant. So the recombination will result into the formation of recombinants. And these recombinants show great variability due to the combination of genes. So here we see that the linkage groups is actually all the genes which are present on a chromosome, and depending upon the position of the genes on the chromosome, they can be inherited together or they can be passed upon separately. So if they allow, that is what is we are talking of linkage. If they do not allow crossing over, then they are said to be completely linked, whereas if they allow certain degree of exchange of genetic material, that is known as incomplete linkage. Recombination and crossing over are important from the point of view because we come to understand the variability which is seen in population. In the next video, we are going to talk about Morgan's contribution in our understanding of linkage and recombination through his experiment on Drosophila melanogaster.